Hello, everyone. Hello. Happy Wednesday. Happy March. Thank you all so much for joining us today. Welcome to FAU Harbor Branch Oceanographic Institute and the John and Barbara Ferreira Ocean Science Lecture Series. I've met many of you over the course of the season, but my name is Dr. Gabby McHenry, and I'm our Director of Education and Outreach here. So a very warm welcome to everyone today. As always, I'd like to begin our lecture by extending a very special thank you to everyone who supports our programs here, and especially those who have made a gift to support our Ocean Science Lecture Series, which many have this season. So thank you so much for your contributions. It means so much to us. There are many great ways that you can get involved and make an impact here on the Institute, from, of course, participating in our programs to sharing them with your family and friends, or making a gift to support our research and education programs. And if you'd like to learn more about that, I'd be more than happy to chat with you after the lecture. I also, of course, want to thank our staff and our speakers, because the series wouldn't be possible without them. And so you've all been attending many lectures, so you know the drill at this point, but we're entering our last month of lectures. Our season runs every Wednesday through March 26th. As a reminder, your registration is good for all of these, so no need to sign up again, but there is still time to encourage your family and friends to sign up to join us as well. And of course, each presentation is also available virtually, so hello and welcome to everyone who is tuning in on our live stream. And if you miss a lecture or you want to go back and watch it again, you can find it on our website or in the emails that Brandon sends out each week. And for those here in person after each lecture, we host a meet and greet reception with our speakers in the gallery and cookies. So we hope that you'll join us for that. Take advantage of our pop-up gift shop and also take some flyers to learn more about our public education programs and opportunities here. So next week's lecture is on March 12th, and that is titled Exploring Rich Natural Product Chemistry of Florida's Coastal Waters by Dr. Amy Wright, who's a fantastic speaker, so I hope that you'll all join us for that. And today's lecture is a little bit different, actually, and it came about from a suggestion that we received last season, so this has been in the works for a little while. We had two separate longtime lecture attendees come to us with this idea, so great minds think alike. They were interested in learning from the next generation of scientists, our students. So here at FAU Harbor Branch, we have 60 students either on our site, working alongside our research faculty, either on their master's or PhDs. And they're doing some amazing work, and you'll hear about that firsthand today. And so I also wanted to take a moment to say, if any of you have a lecture that you really loved this season or an idea of a topic that you want to hear more about in the future, please let me know. I'd be happy to try to work that into the lineup. We value your feedback and would love to do that. So today's presentation is titled Coral Reef Connectivity and Conservation in a Changing Climate by two outstanding students. Our first presenter, Brian Eckert, is currently a PhD candidate in the Integrative Biology doctoral program at FAU and a member of Dr. Joshua Voss's lab at FAU Harbor Branch. He completed his bachelor's in the honors program at Florida State University and worked for NOAA's Flower Garden Banks National Marine Sanctuary before joining FAU Harbor Branch in 2017. After completing his master's degree with Dr. Voss, Ryan was selected as an FAU Presidential Fellow, recognizing him among the most talented students across all FAU doctoral programs. He's also been awarded 2 independent Patty grants and was named Best Student Presenter at the prestigious Mesophotic Gordon Research Conference. Ryan is also incredibly prolific in his research here with an outstanding 20 publications to date, nice job, Ryan, including 3 as lead author from his thesis and dissertation research, and a recent featured cover page manuscript in Heredity. His diverse and collaborative research efforts have focused on understanding how deeper coral reefs are connected with their shallower counterparts and exploring population genetics and symbiotic relationships. This work aims to inform conservation strategies that protect coral reefs across Florida and the Gulf of Mexico, fostering a greater appreciation for the complexity and importance of these ecosystems. And he is on track to complete his PhD very soon, so look out for his defense seminar this summer. And our second presenter, Klein, is a second-year PhD candidate in the Voss Lab here at FAU Harbor Branch. She's also in FAU's Integrative Biology program. Allie earned her bachelor's degree from Roger Williams University in Rhode Island with a double major in marine biology and environmental chemistry. She then was recruited to FAU Harbor Branch and completed her master's degree with Dr. Voss in 2022, evaluating the role of genetic lineages and algal symbionts in stony coral tissue loss disease susceptibility in Florida, which was just published in 2024. Allie's dissertation research is focused on thermal tolerance in corals and identifying resilient candidates for large-scale transplant and restoration efforts. As part of a new collaboration between FAU Harbor Branch and the University of Hawaii, funded by DARPA, she's investigating how environmental conditions influence coral survival and resilience to enhance these strategies. Allie's an active member of FAU Harbor Branch's technical diving team and has been awarded several prestigious fellowships from the Women Divers Hall of Fame, American Academy of Underwater Sciences, Zale Perry, and Divers Alert Network to support both her professional development and her research and restoration initiatives. And finally, Allie is a dedicated and gifted student mentor, including tutoring more than 400 undergraduate students and serving as an instructor for our junior scientist program in partnership with Indian River Land Trust. So what we're going to do is the students are going to co-present today. So Ryan will speak first, and then Allie will present. And following their lecture, Ryan and Allie will be joined by Dr. Josh Voss here on our stage for questions as a panel. So please hold your questions to the end. But with that, please join me in thanking our speakers and welcoming them.
Hey everybody, I'm Ryan, and as Gabby said, I'm a PhD candidate in Josh Voss's lab. I don't know that I can live up to all the hype, but I'm going to try. So I would like to say it takes a village to raise an idiot such as myself, so all of this was not done in a vacuum, and I could not have done anything that I've done so far without tons of help from lots of people, including all my co-authors, on all the work I'm going to present today, everyone in the lab that's helped previous and present times collect data for all this work, the crew from the RV Manta at Fire Garden Banks National Marine Sanctuary. All of my sequencing is done at UT in Texas, and all the computational capacity for these results was done with the help of FAU Research Computing Services. And I really want to point out that a lot of this work, it wouldn't be here at all, and I wouldn't be here without really important funding from NOAA. So in our lab, we focus mainly on corals, and we know a lot about coral reefs and how important they are, but I think sometimes we think mainly about the coral host when we talk about corals, but it's just important to know that they're also this really complex series of symbiosis between the coral host itself, the animal, and then the endosymbiotic algae, dinoflagellates, and then also this whole consortium of bacteria, viruses, and archaea, fungi, etc. So this is the coral microbiome, but mainly what I do is I work on the coral host and then sometimes their symbiotic relationship with algae. Throughout the whole world right now, coral reefs are in a rapid decline, and a lot of this is somewhat from natural threats such as hurricanes, which we're pretty familiar with, especially after last year. And then there's a lot of coral diseases that occur on reefs naturally, but these can also be impacted and increased by anthropogenic inputs, and there's also just standalone anthropogenic threats such as overexploitation, building, coastal development, pollution, and of course raising sea surface temperatures, which are very detrimental to corals through bleaching, where they lose their endosymbiotic algae and turn white, and if they don't recover, they'll eventually perish. So, as Gabby said, I work a lot on deeper reefs, and we call these mesophotic coral ecosystems, and that's just from the word meso meaning middle and photo meaning light. So these are deeper than shallow reefs, typically 30 to 150 meters below the sea surface, and at at that level, we start to lose some of the downward irradiance of the light. So it's darker and less spectra are getting down to those depths. So they also not only have different lights, but they have different temperature regimes. But these ecosystems are also very biodiverse, and they have a lot of habitat complexity. Some of the differences that we might see between mesophotic coral reefs and shallow coral reefs are the differences in the coral communities that are there. There's different species, different makeups of species. And then sometimes we see differences, shifts in the endosymbiotic algae that they associate with at those depths, where there's a difference, and they're maybe selecting for algae that do better at lower lights and for photosynthesis. And then for my research, it's important to know that there's approximately 25 to 40% overlap in the species that occur both shallow and deep, because I'm interested in looking at the connections between the populations of the same species, deep and shallow. So historically, there's not as much research on mesophotic coral ecosystems as other coral reef research. So up until 2008, there was only about 357 publications. But to date, I work sometimes with mesophotic.org, and we put out a paper analyzing all of this data that we've collected. And until 2024, we found that there was now 1,500 publications, or a 4.2% increase throughout those past several years. Part of this might be because data collection is now becoming easier, cheaper, and safer with advances in diving technology and underwater robotics. But even though it's an actively growing field, we still have a lot of need to better understand a lot of these differences between these deep and shallow reefs. And so I would urge you, if you're interested, to check out mesophotic.org. So a lot of the work that I'm doing is kind of formed by this deep reef refuges hypothesis, where there's the idea that maybe deeper reefs might be more sheltered from stressors such as temperature, wave action, human stresses, anthropogenic inputs, and that these deeper reefs, they might be able, if they're staying healthy and reproducing, that they might be able to reseed shallow reefs with their offspring. So even if the shallow reefs are being degraded, the deeper reefs might be able to replenish those populations. But for my research, we're really interested in how can we measure or estimate the levels of connectivity or similarity between these populations across these depths. And I would like to point out that we know this isn't a universal truth. There's a lot of research that shows that there's variable reproductive outputs between deep and shallow reefs. Deep reefs are simply just not universally shielded from all of these inputs and negative effects. Like, there's lots of evidence of big typhoons damaging these deeper reefs still. And we know that the connectivity between regions, it varies across different regions, and it varies within some regions by the species. So there's lots of moving parts to this. Our lab has done a lot of work previously in the northwestern Gulf of Mexico and also throughout Florida and parts of the Caribbean. Lexi Sturm and Michael Studevan, this is a lot of work that they've built up and I've built upon going forward. So with Montastria cavernosa, we looked and we found throughout the Gulf it's predominantly a single population, and there's lots of different populations throughout the region, and there's these complex sort of levels of connectivity and lacks of connectivity across depth and across space. My research, I said, is heavily funded by NOAA. You've heard a lot about NOAA recently, probably. So this was part of the CYCLE program. So we were interested in looking at this connectivity of ecosystems throughout the northwestern Gulf of Mexico, looking at octocorals, black corals, sponges, and stony corals in terms of the benthic species. And right now, I work on the sponges and the stony corals. So I said my lab, in Josh's lab, we work mostly on corals, but I'm kind of an outlier, and I, for some reason, decided to work on sponges, which they're a little difficult at times, but I still love them. So like I said, I'm interested in knowing the connectivity. So I'm interested in looking at how similar are populations of the same coral species, so across sites in the northwestern Gulf of Mexico, but also across depth within the same site. So how can we answer these questions? Well, I do it through the use of DNA. And I would just like to kind of quickly go over like what we do with DNA. So this is me, and you might think that this guy looks a lot like me, and you wouldn't necessarily be wrong. But it's kind of interesting that depending on how you calculate it, me and this chimp share approximately 98% of the coding regions of our DNA. With a mouse, it drops down to about 85%, but that's still a passing grade, and that's pretty crazy to me personally. And that's actually the reason why we can use mice in a lot of genetic studies to inform medicine and so forth. And then just for fun, not super relevant, but 60% with the banana. It's kind of this, and this is indicative of, I think, what would be like if it ain't broke, don't fix it. So there's lots of kind of similar protein codings and stuff that it doesn't matter. There's no reason to reinvent the wheel because we share similar needs even with the banana. And then just for fun, me and the GOAT, LeBron James, I know what you're thinking. Are these guys twins? Maybe. We share 99.9% of the same DNA. If that was $10 in pennies, the difference is 1 penny. So all that is to say, it's not really about what we share, it's what's different that makes us unique. And so we can kind of leverage this to find out the differences between these populations to see how similar or dissimilar they are. And differences in DNA can come about through mutations. Not quite like this, but this is, from the fly, so there was a change in the genetic code, and then this was like a drastic mutation, but mutations can be random, they can be deleterious, where the organism just perishes, and they usually happen through evolution through thousands and thousands of generations. So all this is to say it changes the genetic code. And what I do is I use single nucleotide polymorphism, or SNPs is how we just pronounce it, SNPs. And this is a way where we can see in 2 different individuals within a population, at certain points in the genome, there's a mutation. So this is the same sequence, but within this same sequence, there's one base pair that's different, and we can leverage that and look at the genetic diversity of these populations through these SNPs. So to the actual work that I've been doing, in the Gulf of Mexico, we have 5 sites throughout what's Flower Garden Bank's National Marine Sanctuary. And I'm working on 3 coral species, Montastraea cavernosa, Stephanacina intercepta, Orbicella faviolata, and then the renegade sponge over here, Cestospongia muta, bane of my existence presently. So we collected these using some various technical diving, using open circuit scuba, closed circuit rebreathers, and we collect small samples and we preserve them, not all into one tube, but for these purposes it's going into one tube. And we keep those samples and we extract their DNA so that we can target those SNP sites throughout the genome. So diving into some results, I have these split by species. I've tried to color code it and have a little picture just for your reference. And we're going to see the same things over and over a few times, so I'll walk you through this one a little slower. So these are structure plots or admixture plots. So each color here refers to a distinct genetic lineage. So that's kind of like an ancestry, like you would do through 23andMe or something. So there's 2 kind of ancestral populations or genetic lineages of Montastraea cavernosa in the Gulf of Mexico, based on these data. So the amount of each color in each of these individual lines, each line is a colony, and they're split up by shallow and mesophotic in each of the 5 sites that we sampled. So it's the proportion of membership to each of those lineages. So when there's one that's all this dark green, that's an individual from that line. If it's all the light, that's an individual distinctly into that line or lineage. And then there's mixtures in between. There's interbreeding, back crossing, and so we get these, in this case, we get these kind of Kleins of admixture or hybridization. So in MCAV, we see 2 distinct lineages. And if we look at where those lineages fall across depth, all of this shaded area is in the mesophotic zone. Our first smaller cluster here is it's all exclusively in the mesophotic zone, and then the other one is found both shallow and deep. And then the hybrids or the admixed individuals, they're all over the place, but they are more heavily in the mesophotic areas. So, and then if we looked at this, we can see that there's a lot of similarities between banks. So East and West Bank both have kind of similar proportions of the ancestry in MCAV. I'm also interested in not just the population structure, but the levels of genetic diversity. So using those same SNPs, we can look at it and we see there's really not much difference in the genetic diversity. So this is heterozygosity. It's a measure of genetic diversity for all intents and purposes. And so we see that there's really not a big difference between the two lineages. The admixed ones are slightly more heterozygous, which is kind of what we would expect from crosses between these two. And then this measure FST between these, this is a measure of genetic differentiation between populations. And if it was 0, they would be completely identical in terms of their genetic makeup. And if it was 1, they would be so crazily different. Like, I'm not even that far from a banana, apparently, right? So we have to know that 0.04, it's moderate, but it's still fairly low. So Stephanocina intercepta. This is a little different. We still find likely 2 lineages, but there's a lot of hybridization. We have a few individuals that assign to the dark cluster and a few individuals that assign to the light cluster, and there's a lot of hybridization between them. And just looking at these, you can almost see, like, it looks almost the same throughout every depth and every site. And that's verified if we look at the distribution across depth. There's really no difference between these statistically. So I think that these are functionally one single population of interbreeding genetic lineages. And again, no differences really in this species across site or depth. So in the genetic diversity, it's similar. It's actually, there's very low differentiation between the two lineages, and again, similar levels of genetic diversity among them, and then slightly higher in the heterozygous, or the admixed individuals, or more heterozygous, excuse me. So Orbicella favulata, the mountainous star coral. In this, we see 3 distinct lineages, and these are pretty heavily partitioned across depth. So we have this first deep lineage that's mostly found deep, a second smaller lineage that is exclusively deep, and then almost all of this third lineage is found shallow, except for one little guy that made it down and stayed alive. So that's also, there might be some sort of settlement issues here where there's some kind of environment and phenotype mismatch, but we don't really know at this point. But these are lines of future study that I'm very interested in pursuing. And again, we have very similar structure between East and West Bank, which I typically would expect to see because these are only about 10 nautical miles apart from one another. So they're very close. In terms of the diversity, we see that there is some slight differences, but overall they're relatively the same levels of heterozygosity as all of the other coral species that I've just looked at. And it's interesting to note that the deep lineages, so these two here, the first two, are much more similar to one another than either are to the shallow lineage. So that, I think that that's interesting, and that might indicate some level of recent divergence among these lineages in terms of evolutionary time. So Zestispongia muta, this is part of the reason why it's been a pain, is that it's much crazier. There's at least 6 lineages that we can find in this data. There's tons of depth partitioning. These 4 deep lineages are exclusively deep. And then we have a lineage that's all over the place and one that's mostly shallow. And pretty much every bank is distinct from one another, and every depth, when we have both depths at a bank, are distinct from one another. So everywhere that the Zestispongia is in the banks we've sampled at Flower Garden Banks, it's its own kind of unique population, which is cool, but it's been kind of hard to deal with sometimes computationally. And so there's a lot of variability in the diversity between those lineages, much more than in the corals. And overall, I keep finding, and it's not crazy, I guess, that there's a lot lower diversity as compared to the corals. Zestispongia, when it spawns, its eggs are negatively buoyant, so it doesn't maybe have as much opportunity to disperse, which means you're having more closely related individuals within a population, which leads to higher rates of inbreeding, which lowers genetic diversity. So that's kind of my hypothesis of what's going on here and some other future lines of research to look into. Because Josh, I'm graduating hopefully, but Josh isn't getting rid of me anytime soon. So kind of just to wrap it all up quickly, coral and sponge populations throughout flower garden banks have a lot of variability in the levels of connectivity and divergence within those populations. So it varies by species, and then some species have no connectivity across depth. Other species have a lot of connectivity across depth. So studies like this, well, a lot of the reason that I'm interested in this is to help enhance conservation and management strategies. So looking at these data, we can kind of pinpoint, in some instances, which populations are bigger sources, which should be prioritized for protection if we only have so many resources to attribute out to populations. And then we can use this in some of my other work that focuses on other sanctuaries and other regions. We can use these data to see how similar the populations are across regions, and ideally would be able to develop collaborative efforts, or what we would call sister sanctuaries. So we have the same individuals or animals, and we have very similar populations. Mine feed yours, yours feed mine potentially. We need to work together because it's all really one big population in the end. And so, like I said, I'm also working presently, wrapping up, doing this in other MPAs in Florida, up here near us in Kristen Jacobs Aquatic Preserve, and then down in the Florida Keys National Marine Sanctuary. So, like I said, it's very important to me. NOAA has funded all of this research that I presented, and pretty much my entire career since 2009. I'm been around for a while, so that's why I think it's had such an impact on me personally. But I would, if you're interested in this or you think it's good, these are the people to reach out to. They're supposed to represent our interests, but they don't know what we're interested in unless we tell them. And so I would urge everyone that feels compelled, if you think that we shouldn't be maybe cutting all this scientific funding, to let everyone know and let them know where you stand. So that's it for me.
Well, thank you, Ryan. Ryan is always a tough act to follow. But my name's Allie, as Gabby introduced, and I'm going to take us about 4,000 miles away from the Gulf of Mexico to the Hawaiian archipelagos, which is where my dissertation research takes place. So in case anyone hasn't had the benefit to get out there, Hawaii is at the northern limit of the coral distribution in the Indo-Pacific. This is buzzing. The Indo-Pacific is a hotspot for coral reef ecosystems. It has about 700 coral reef building species, whereas here in the Caribbean we have about 80. However, Hawaii is biogeographically isolated, as you can see up here. So it's pretty far away from all those other reef systems, which has led it to have very unique research abilities. There is high levels of marine endemism in Hawaii, meaning that there are species that occur only in Hawaii and nowhere else in the world. There's about 60 stony coral species in Hawaii, so a lot less than that of the entire Pacific. And they're mainly dominated within these three genera, the Porites, the Montipora, and the Poeciloptera. However, the reefs of Hawaii have some of the most coral cover in the world. They have about 30 to 50% coral cover on all of the main Hawaiian islands. To give you reference, here in Florida, we have about 13% coral cover. And Hawaiian reefs offer a lot of importance to not only their native Hawaiians, but also all the people who live there as well. So one of their main importance is they have high cultural significance. In the Hawaiian creation chant, the coral polyp is the first individual to emerge. So there's this really deep spiritual connection with natives to the ocean. They also serve as vital food resources for local communities and locals' livelihoods. One of their main fisheries habitat organisms is their tuna. And also the reefs themselves offer other activities such as surfing. Surfing is really big in Hawaii. When I was out there, I actually got to see some of the Olympic trainers train out in Hawaii, which was cool. And then they bring in over $400 million in marine tourism. So this can be anything from snorkeling to scuba diving to fishing charters, all of the ideals that draw you to Hawaii. And lastly, one of the things that reefs offer is coastline protection, and this often goes overlooked, of not really understanding how coral reefs can protect our coastlines. So to give you a reference, coral reefs can dissipate up to 97% of wave action, which is really impactful when we have things like hurricanes or other storm surges that may impact our coastal communities. Us here on the Treasure Coast know how much people love to live by the beach, given its beauty. But those communities can be really impacted by things like flooding, hurricanes, or any type of storms. So a study was done recently in 2021 to actually put an economic value on what these reefs are doing to reduce this flood reduction. In Hawaii, reefs are valued at about $800 million a year. So this is what the reefs provide to those local communities in terms of flood risk reduction. On the island of Oahu, which is where my dissertation takes place, they have about half of that. So $400 million a year annually is how the reefs protect their coastlines. And although reefs are culturally significant, they have a lot of ecological benefits that Ryan had mentioned in his talk, and they have these coastal protection importance. They are still, unfortunately, being threatened worldwide, as Ryan had mentioned. One of the threats that I focus on in my dissertation research is the idea of coral bleaching. And so to understand coral bleaching, we have to understand the relationship between the host and their algal symbiont. And so this is just a small little schematic to show that corals harbor these symbionts within their tissues. The coral provides a home for the symbiont to live free from predators, and the symbiont, in exchange, gives the products of photosynthesis, so it gives energy to its coral. Caribbean corals, as well as Pacific corals, can get up to about 90% of their energy from these algal symbionts. So what is bleaching? Bleaching is a cause of dysbiosis or dysregulation in that relationship from some type of stressor. For my research, I look at thermal stress. So prolonged increased sea surface temperatures cause the corals to expel this algae and reveal this stark white tissue. The algae is what gives the coral its color. And so this is fine for a couple of days or hours, but the coral is slowly starving. So if this bleaching continues or if this increased stressor continues, the corals will eventually starve as they no longer have that 90% energy and die. And this is when the skeleton becomes overgrown with algae, and this is where we start to see those phase shifts in coral-dominated reefs to algal-dominated reefs. Mass bleaching events have occurred throughout the decades. About a couple of years ago, we had a major one here in Florida that impacted the majority of our reefs. One of the most notable bleaching events that happened in Hawaii occurred in 2014 and 2015. They saw about a 71% reduction in their coral cover on the west coast of Oahu, and about 50% reduction in coral cover in this place on Oahu called Kaneohe Bay. And this is a widely studied bay environment. This is where a lot of my research is being taken place. Kaneohe Bay is some of the highest coral cover in Oahu, and so when they saw this much reduction, it really caused scientists to put a lot of money into researching that bay as a whole. A subsequent bleaching event happened in 2019 that was less severe than the one in 2014, but still pretty impactful, as about 30% bleaching was recorded on Oahu. And what was noted is within Kaneohe Bay, they saw that some corals were experiencing a variable response to this bleaching. So as you can see in this picture, this is of a coral species, Montipora capitata, which happens to be one of the species I work with. It's actually endemic to Hawaii, so it's only found on Hawaiian reefs. And this is a picture from 2019 where you can see this is a bleached coral, and of the same species, this coral next to it remains unbleached. So this promotes the idea that there is some level of thermal tolerance occurring within the species that's variable. And so scientists have asked the question of what makes a coral thermal tolerant, even though they're the same species, and how can we leverage these corals that are thermally tolerant when we talk about restoration. And we want to restore these reefs with resilient corals that will be able to withstand future ocean conditions. So this brings me to the parent project of what my dissertation is kind of umbrellaed under, called the Rapid Reef Resilience for Coastal Defense. This is a project partnered with the University of Hawaii, Hawaiian Institute of Marine Biology, University of California, San Diego, Mackay Engineering, and us here at Harbor Branch. And the idea is to create this living reef structure that's main goal is to dissipate wave action. However, as scientists, we know that how much effort goes into something like this, like a reef restoration project. Not only time effort, but money effort. And so the idea is to restore a reef with resilient corals so that you no longer need scientific or human intervention once that reef is installed. So we've come up with some other ways on how to make this reef sort of the best way to restore this area. And so this comes with adding micro-architecture within the reef to call for recruits, so other corals from the native population to come and reseed on that reef. There's some engineering going on to look at light-enhanced heterotrophy, so different types of feeding. And what my research was focused on is selecting the best corals to go out on this reef, or selecting for favorable traits, in this case, corals that are thermally tolerant. And so the main pilot for where this reef structure is going is a place called Kailua Bay. And so this is to orient you. Again, this is the northeast side of Oahu. This is Kaneohe Bay, which is the widely studied bay with thermally tolerant corals. And this is Kailua Bay, just over this peninsula. And so similar work to what Ryan had done, other researchers had done in the University of Hawaii, and they found that these two populations are very well connected genetically. So it is a good idea to bring corals of the same species from Kaneohe Bay to Kailua Bay because genetically they'll be able to reproduce with one another. However, these two bays are very different environmentally. So Kaneohe Bay is the largest sheltered embayment on Oahu. It has, again, some of the highest coral cover on all of the Hawaiian Islands at 60%. And due to the water flow and the different dynamics within the bay, it retains high ocean temperatures. It's relatively shallow, and that warmer temperatures year-round have caused this acclimated thermal tolerance within those corals, as we saw. Kailua Bay is a lot less nice to dive in. I would explain Kaneohe Bay as a bathtub and Kailua Bay as a washing machine. It is much more similar to open ocean conditions, so much higher wave action, lower temperatures because you have that increased water exchange, and also the bottom of the reef is about 30 feet, whereas Kaneohe I averaged about 15 feet. So then you have differences in light and sedimentation and all of that as well. But again, they're connected genetically, so we still think that it may be a good idea to select corals from this one bay and bring them to the other. And this is also largely due to this Kailua's Bay, or the Ulupau Reef, has been largely degraded. So it doesn't have a lot of high coral cover, making this a good site to put this outplanted reef project to help add some of that three-dimensional complexity back to the area. So this is my map of just where I'm collecting my samples from. I have 3 collection sites with Kaneohe Bay. So again, the idea is to select corals that are going to be best put onto this structure. So I wanted to get from different populations within Kaneohe Bay to sort of see where we should source them the most from. I also have a collection site from Ulupau, or Kailua Bay, and this is meant to serve as a native control. When we do these types of transplanting studies, we always want to compare to what our baseline is within that native ecosystem. So how are native corals growing and thriving within that ecosystem? And that can serve as our baseline when we take corals from a different habitat and bring them over. I have 2 outplant locations. One of them is back in Kaneohe Bay. Again, this is serving as the same control to see how quickly these corals are growing and what's their overall survival rate in their home environment. And then my experimental site is in that Kailua Bay where the reef structure is going. I selected 2 different study species with 5 genotypes of each species across all of these 4 sites. Montipora capitata, again, the endemic coral species that has the variable thermal tolerance, as well as Paredes compressa. These 2 are the main reef builders out on Oahu. This is actually a picture of me collecting Paredes, and you can see these are all Paredes colonies. So large coral cover. They cover the entirety of the reef, and they really contribute to that 3-dimensional complexity. I have about 40 genotypes total, And once I collected them, they were cut up into fragments and glued onto these coral plugs to be able to be outplanted. This is just a timeline of how and when this work was done. So the original colonies were collected from the sites in November of 2023. This past May in 2024, they were able to grow up during those 6 months, and that's really just so when we outplant them, they're skirting over that plug and they're more sturdy for that transfer. But of my 8 plugs, I had 6 of which go towards the transplantation. So 3 are going back to Kaneohe Bay as that nursery control environment, and then 3 fragments per the genotype is going to Kailua Bay as part of the experiment. And then I had 2 extra fragments per genotype that are going to serve as my initial thermal performance test. So what is the natural thermal tolerance of these corals prior to any transplantation? And so how I did this is they underwent a stress test where the corals were put into a water bath and ramped up 1 degree Celsius per day and held for 16 days. And so this was to test how long it took for the coral to show some type of thermal stress. These were done in tandem water baths. Those are my 2 fingers showing you in case you couldn't see. And the experiment was run by this controller. And so what was done is as the temperature ramped up throughout the weeks, I used this really cool instrument called a PAM fluorometer to take a picture of the coral and calculate that photosynthetic activity of the algae. So this essentially provided a baseline of what the photosynthetic activity was prior to any heat stress. And then as heat stress went on, I was able to see how that photosynthetic activity declined. So on this figure, we have our photosynthetic activity. At 1, this is what it started at, and each of these points is each of the days I sampled. So the slope of this line is showing a steep decline over time to show how the coral is slowly bleaching or how thermally tolerant the coral can be. And so this is all measured in degree heating weeks. And degree heating weeks is a method used by NOAA to sort of predict bleaching on a various reef. And what a degree heating week is the number of weeks a coral experiences temperatures of 1 degree above its mean monthly maximum. This is a photo from 4 different years. So this is 2009 and 2010. These are the Hawaiian Islands. And then this is the 2014-2015 event where you can see that they had a lot, about like 10 to 12 degree heating weeks across those islands. So that was when the water temperatures were really warm. 4 weeks has been calculated as the number to predict coral bleaching on an area. And 8 weeks or more than 8 weeks degree heating weeks is leading to wide-scale bleaching events and mortality. So the results from my initial thermal performance test showed variation between species. So Montipora capitata had different thermal tolerance than Parietes compressa. Parietes compressa actually had high thermal tolerance, and it's measured here on the y-axis in our degree heating weeks. But I don't want this to fool you because both of these species have above that 4 degree heating week line, so they're both pretty thermally tolerant species. But the goal of my study was to really tease apart where we should source these corals from when we're putting them out on that large structure. So I broke them up by collection site. So I have 2 panels here, one being for the Montipora species and one for the Parietes species. They do have different y-axes, but they're all in degree heating weeks. And what this graph is showing is that Parietes compressa, from among their collection sites, they show greater variation in their thermal tolerance. Especially in this the Kaneohe Bay cooler site, or one of my sites in Kaneohe Bay, this blue for both of those sites, they have above that 8-week degree heating week thermal tolerance. So these would most likely be some of the candidates that I would select when putting out on that artificial reef structure. That is, if they are surviving the harsher conditions of Kailua. So this brings me to the transplantation part of this study. So while these corals were in the water bath ramping up for the 16 days, I took other fragments from that same parent colony and put them out onto the reef. And so this was engineered to go right down on the reef structure Again, all genotypes were present, and they were left out at the Ulupalu Reef and only monitored every couple of months. At the 6-month survivorship, we found that about 52% of our Porites corals were still alive. Now, this may look like a failing grade, Brian would say, but this is pretty comparable to other reef restoration studies where you do lose about half of your initial transplants. However, this is a lot less than the corals that were outplanted at the control site in Kaneohe Bay. Montipora, however, dropped down to only about 30% survivorship after the 6-month period. I should mention that although the species Montipora capitata was present at both Kaneohe Bay and Kailua Bay, they are existing in different morphologies. So in Kaneohe Bay, they're more of a branching or tree-like morphology, whereas in Ulupalu or Kailua, they're more plating. And so this might be just an adaptation over time due to the high wave action that these branching corals might not necessarily be suitable for that habitat. And so the next part of my work brings me to actually going back out to Hawaii in 2 weeks to run this second thermal tolerance, because now I know that which corals were the most thermally tolerant before transplanting. I know which corals survived this first 6 months of transplanting, or now it's about 9 months. And so now I want to see, kind of interjoin the two, which corals are surviving and which corals are also maintaining that thermal tolerance. Again, these two bays are drastically different. So corals that were once used to the conditions of Kaneohe Bay may be different or may change their traits when in Kailua Bay. So the second thermal tolerance test is to see if that thermal tolerance has been maintained over time throughout transplantation. So some of the projected outcomes for my work are to first identify which location and coral species exhibit the highest level of thermal tolerance mixed with their survival. So again, selecting for those sort of double action traits for this outplanting project. Then, is thermal tolerance retained within genotypes, or are the corals acclimating to this new cooler condition? And all of this is to help guide the outplanting strategy to select resilient genotypes for these future climate change scenarios. Other parts of my PhD research are looking more at some of the molecular changes or molecular indicators of this type of thermal tolerance, so looking at gene expression profiles. I also took initial symbiont measures to see if different species of symbiont may be driving their thermal tolerance, as well as I have been measuring them quarterly to track growth over time. And why this is really important is, again, when we're putting corals out on a structure like this, we, one, want to make sure that they survive their new conditions. We, too, want to make sure that if we're selecting for something like thermal tolerance, that this thermal tolerance will hold once they're in that new condition. And three, it doesn't really do us much good if the corals aren't growing and thriving in that new environment, given that the growth and the morphology of a coral is really what contributes most to that three-dimensional complexity, which helps dissipate the wave action, which was the main goal of this project in the first place, is to create a reef to dissipate wave action. So when we were talking about restoring or restoration, we really like to drive home this point of restoring with resilience. What is going to make this reef successful and sustainable for future generations without constant human intervention? And so with that, I would just like to wrap up by, again, echoing what Ryan had said. None of this happens in a vacuum. I would like to thank my advisor, Joshua Voss, as well as Ford Jury, who's my main advisor out when I'm in Hawaii, as well as my lab members and all the lab members of the Coral Resilience Lab who sort of adopt me when I'm out there. And just would like to highlight two of my past mentors who have recently passed away, who were really inspirational throughout my academic work. And so I'm just going to leave this slide up as Brian and Josh come up and help me answer questions. There is a QR code, which is if you want to help donate to our lab or if you're interested in our research, you can scan that. I'll have them come up.
So I basically just I'm basically the buffer. If there's any questions that you all ask that are too hard, I'll defend these two to the death. But really, I would encourage you to ask questions of them, not questions of me.
Yes. So the question is, when do we decide to go back out to a reef to collect our studies, and what factors do we consider? And so for my particular study, it really depends on the questions that I'm asking. So we designed this project to go out every month and monitor. Every 3 months, I would fly out to Hawaii and do monitoring. However,
We both were.
yeah.
Then the budget got cut, so I don't get to go.
Yeah. So funding is a large part of it. Weather is another thing. So getting out to a site repetitively is pretty hard given weather and boat availability and being able to actually dive these sites. In the case of my work where I'm deciding to go back out in March, is I'm really interested in this question of conservation of thermal tolerances. Is this trait conserved after they're in this climate refuge or this deeper environment? And I need live coral to do that test on. So as I saw that my corals were dying, I wanted to go back out and of the ones that are surviving, test that thermal tolerance.
Same. For me, I'm mostly interested in sort of that ancestry of these populations. So my goal is to get as many as I can from as many places as I can whenever I can, because we're kind of looking at the ancient makeup of these populations in a way. But to your point about catastrophic events and stuff, my data right now can serve as a really good kind of baseline, and then I can actually go in and resample populations following some kind of big event if we wanted to, and look at, are there shifts in that population now? Have we completely lost certain lineages? And yeah, so that would be like when I go sample, typically for what I'm doing, it's just when the weather's good and we have that funding to get there. But I could repetitively go after big storm events or something and see what happens moving forward.
So relative to that, Ryan's also done work on one of those same species in the Florida Keys that since got completely dramatically impacted by both the 2023 bleaching event and by stony coral tissue loss disease from about 2020 through 2024. We just put in a proposal with a new faculty member at FAU, Karsten Grupstra, to go back and look at those same populations and see how have these events changed how those populations look, and does that change our restoration plans for the Florida Keys as well?
Again, so that's one of the things that we're looking. Oh, sorry. The question is, what is the future of coral reefs given the increasing water temperatures? And so that's what a lot of the work out at the Marine Station at University of Hawaii has, where they're looking to see these corals that have already sort of micro-adapted to some of those future ocean conditions is what we're calling it, within Kaneohe Bay. So to give context, the ocean temperatures at Kaneohe Bay are about 2 to 3 degrees Celsius higher than the surrounding bays year-round. So we look at it as a way as they're about a couple decades or faster than us. I don't know if I explained that right, but they're ahead of us in terms of this overall climate change. So we're looking at to see how these corals are adapting, and then we could look at things like biomarkers for corals that may be thermally tolerant. And then when we talk about these restoration projects, using those corals that we know have these traits to outplant on the reef. Now, when talking about coral restoration, there's only so much we can do. It is sort of a Band-Aid on a bullet hole in some instances, because there's eventually a threshold where a coral will give up. So we're just hoping to not reach that thermal threshold. But when we restore, it's again the idea of restoring with a scientific background of selecting for traits that are more favorable for these future ocean conditions. And again, connecting to some of Ryan's work of these like deeper reefs that may be able to reseed the shallow populations that get wiped out by some of these wide-scale bleaching events.
Do you want to talk about lack of these problems on deeper reefs?
Well, a lot of deeper reefs have these problems to a lesser extent. So there is this like idea of hope from these deeper reefs, but I would also say that, like I said, there is a lot of impacts to these reefs still that we're finding, and in a lot of cases, there's reduced overall like species diversity on these reefs. So while they can serve to help be source populations, they're not the answer. And I think that the real issue is, like Ali kind of said, we're treating symptoms rather than causes. Because like restoration is great, but it's not an answer. It's like a temporary Band-Aid trying to help, but we really need to solve the problems that are causing these rapid declines.
And the only thing I'll add is that particularly here in Florida, like we're moving into a phase where the vast majority of the corals and the coral ecosystem services that we have are those mesophotic reefs because we've lost so much that they're shallow. So understanding how those function independently and advancing how we protect and conserve those systems is probably one of the most important things we can do in terms of fisheries sustainability in Florida and other places as well. So John's asking a question he knows the answer to, since he is the world's expert on oculina reefs. Thanks, John. So corals have facultative symbioses. They are complete mixotrophs, meaning they can be both heterotrophs and autotrophs, and they can go back and forth not just within a day, the way most of them do, where they retract their tentacles all day long and do photosynthesis and get that 90% of their sugar, like Ali was talked about. But then they also, at night, extend their tentacles and feed on zooplankton all night long. And that's probably part of why corals are so incredibly successful, is this mixotrophy approach. What's really interesting for oculina is that within this one species, there's this huge range in what they can do physiologically. And it's not just that the shallow ones have algae and the deep ones don't and focus exclusively on feeding. But John has actually demonstrated that they can switch. You can do transplants from deep to shallow, and as you move them, they will change modes to acquire zooxanthellae if you move them shallow, or lose their zooxanthellae when you move them deep. So some of that, I think, gives us at least some indications that corals are capable of a whole lot more physiologically than we recognize to cope with changing environments. And the better chances we can give them to recover, the greater likelihood that they will. But there are definitely some species, and Brian kind of hinted at this, there are some species that are only shallow. We're not going to find them in those deeper areas to recover. And there's two ways to think about that. The first is to say they're not good candidates. We shouldn't invest time in them. We're going to lose them. Move on. Or to instead say we're going to focus on trying to identify traits within the few remaining individuals of those shallow species to help to try to protect those species. And that's at least being done in one group, the Dendrogyra, the big pillar corals, that have been really heavily hit by disease and thermal stress in Florida. And essentially at this point, all of the remaining dendrodryra that were left in the keys were collected and brought into captivity so that we can try to do like a condor style restoration approach where we identify how to boost their reproduction and how to increase the traits to allow them to return to the environment. But to Ryan's point, until we get a handle on climate change, it's a very uphill battle.
Yeah, I'm going to X-nay the millions and millions of years of history, and I'll pass that down to Ryan maybe. But for this actual, at least my project, this reef structure is funded by DARPA, which is a branch of the Department of Defense. And this structure is actually scheduled to go in the water June of this year. And so my project started, again, November 2023, so about a year leeway to sort of inform which corals to be actually selected to go out on this reef. In terms of scale, this is going to be a fairly large reef structure, definitely the largest that I've heard about. I believe it stretches 30 meters. Is that correct?
It originally was planned for 100. Now it's cut down to 50. But each of them, it's 50 meters wide and almost 6.5 meters tall by 10 meters deep. So it's essentially, 3 times the size of this stage across the bottom.
And you can see in the figure, we're trying to capsulate all aspects of the reef. So you have the reef slope, the forereef, and then the back reef. And all of these structures are going to be implemented with corals on them. So pretty large in terms of scale. I don't know if anyone else wants to tackle the...
Yeah, I think the key to this structure in terms of sustainability is it only works if the corals that we put on it and the natural recruitment that occurs there allows the corals to themselves grow and reproduce. If you have to continually do this, it's not going to work. It's not a sustainable solution. So that's one of the keys. And there's some architecture in there to favor recruitment and to dissuade algal overgrowth as well. And Ryan, you want to talk about the timeline part of things a little bit?
Which timeline?
How many times have we lost corals to bleaching, and can corals bounce back?
I don't remember. I've only been here for a little while. No, but throughout time, corals have waxed and waned. I think I don't remember how many times off the top of my head, but there's been cycles. But I think the issue is that it's happening kind of at a higher rate now than in like geological history. But like, I guess in theory they could bounce back again. But with humans being involved now, the issue is the continued sort of pollution and things that are exacerbating temperature rise. I think like without getting rid of those, there's nothing for them to come back to.
So I would say Ryan's mostly right. I would say taking a very long view, geologic evidence suggests that corals will survive. There's been massive extinctions and recoveries of extensive coral reefs, including most of Florida, once being a coral reef that was then exposed and subdued multiple times. So when we're talking about trying to protect, conserve, and improve coral assets, we're mostly coming at it from the perspective of we want those things for the coral ecosystem services we're getting now in the next 50, 100 years. On the thousands to hundreds of thousands of year time frame, corals are probably going to wax and wane and bounce back and evolution will find a way. But if we want to maintain the coral ecosystem services that we're currently getting, that's where these initiatives are coming into play.
These are cement. So yeah, they're cement, and then there are some other cooler substrates that might go on it, such as like an anti-fouling, natural anti-fouling to dissuade algal overgrowth and promote coral recruitment. But these main structures are cement, very heavy.
They're reinforced cement with modules that can screw onto them, and then the materials she's talking about are actually really cool 3D printed biopolymers that have bioactive molecules embedded into an artificial biofilm that gets printed onto the concrete. One more. I do. So something like that would be the perfect thing to build and establish that you could disgorge all of those cruise passengers onto and let them be there rather than on the natural reefs. So there's several different resorts and cruise companies, including Disney and Royal Caribbean, that are investing in artificial reef construction specifically to be able to put their people there rather than on natural reef structure. As for the free diving fins for scuba divers, it's ridiculous and it's become a weird trend. It's way less efficient. So the easy thing to do in that case is for all the operators to just ban them from their boats. Simple.
All right, everyone. Well, please join me in thanking our presenters and Dr. Voss for joining for the Q&A. We're going to join you in the gallery, so please come and grab some cookies and continue conversation. And don't forget next week's lecture by Dr. Amy Wright on the chemical diversity of Florida. We'll see you all then.